جمال راس البر. It's nice to see you. عمرو هيكلمنا بقى على موضوع الابيجينيتكس في الكدني ديزيز عامة ولا في الاكيوت كدني انجري؟
سيدنا الاستضافه الجميله وكان جميل دكتور ناجي يا سيدنا. انا قابلت حضرتك دكتور محمد دايما تراي تو بي اولويز اكسشينج يعني انفورميشن والعلم مع حضرتك. شباب جميل ومبسوط جدا ونتمنى ان انا اجي تاني ان شاء الله مره ثانيه وثالثه ورابعه ان شاء الله.
طيب
اوكي ان شاء الله. انا عارف الموضوع شكله ضغط بس ان شاء الله هيبقى يعني محاضره سريعه وسهله. So I don't know how many months الدكاترة معانا هنا is oriented by this topic, the epigenetics, and if it is something that's research study or the applications or not. هنشوف الكلام ده في المحاضرة. Our agenda today is the basics of genetics. What is epigenetics? Basic epigenetic concepts, epigenetic role and applications in nephrology. البداية طبعا كانت في سنة 1865. ميندل كان أول ساعتها واحد فكر في الجينز كنترولينج الانهيرتنس والتريتس، وجه ساينتست كتير بعديه لامار وداروين اتكلموا في الجينيتكس كتير. وكان سنة 2003 مع بداية ظهور الجينوم بروجكت وقالوا ان احنا بنعمل مابنج للهيومن جينوم بروجكت هنقدر نعرف كل الديزيزز وكل الباثوفيسيولوجيز ويبقى العلاج اسهل. لكن كان الموضوع بالسهوله دي والسمبلسيتي دي؟ ما كانش كده، لان في حاجات عندنا مش جاست اكسبلين باي جينيتكس. فور اكزامبل مثلا ازاي ايدنتيكال توينز ممكن يبقى عندهم الوان شعر او الوان عين مختلفه، ممكن نفس الشخص يبقى عنده لون عين ولون عين مختلف. ازاي الستيم سيلز كان differentiate into different types of cells. كل دي اسئله genetics are not enough to explain it. Sometimes scientists explain this او عندهم what known as ghost writers. طبعا مفيش حاجه اسمها ghost writers. It has an explanation. It's epigenetics and not genetics. The first one who explained this or described this was Waddington. وكان في article موجود في التايم why your DNA isn't your destiny. It's not just DNA هو اللي بيحدد who are you and what's going to happen to you. It is your phenotypic variation is controlled by this axis of epigenetics, genetics, and environment. And your genetics is just a tip of an iceberg كبير مستخبي تحتيه epigenetics, the real controller of your DNA, of what's going to happen to you in future. So this rise of epigenetics made a new turning point in the understanding of hereditary as possibly one of the greatest revolution in the century. نظرة سريعة على ال DNA بتاعنا to remind us. احنا عارفين طبعا ان هو is formed of double helix. اوكي هو بيتكون من double helix of polynucleotide, sugar وبيز وفوسفات جروب اللي هو الادينين والجوانين والثايمين والسيتوسين. Its linear DNA has to be packed into a nucleus. والدي ان ايه ده بيبقى coiled around 8 protein core histone يعمل نيوكليوسوم. النيوكليوسوم تبقى coiled وتعمل الكروموسوم. ده سريعا كده ريمايند اس. ده شكل الاوكتيمال النيوكليوسوم والكروماتين والهيستون تيلز اللي بتكون خارجه من الكروماتين. وهنشوف ايه استخدام الكلام ده. So برضه how the genetic code happens برضه ده تذكره refreshing to our memory ان الدي ان ايه بيبقى transcribed into messenger RNA والmessenger RNA through the ribosomes leads to the production of the proteins. This is known as the genetic code. Conversion of genetic information to a protein relies on the transcription of nucleotide sequence of DNA and messenger RNA. طيب السؤال بقى what makes this gene silenced, transcribed, translated, expressed, suppressed? It's the epigenetics. Now we can classify the genetic data we have into first genetic code اللي هو DNA اللي building unit بتاعنا. Genetics based on the DNA sequence اللي موجود عندنا. And then comes the second epigenetic code أو genetic code اللي هو how, when, and where this information will be expressed. اشمعنى في الوقت ده، في الشخص ده، في الزمن ده كان في expression للحاجة دي، وفي وقت آخر أو زمن آخر ما كانش فيه expression. دي اللي اسمها second genetic code. So epigenetics اللي هي معناها what's above or beyond genetics refers to the study of sets of genes. Epigenomics refers to the more global analysis of epigenetic changes across the entire genome. يعني simply كده we are written in both a pen and a pencil. Okay? What cannot be changed in us DNA is the pen, but what can be changed with time is the pencil written اللي هو epigenetics. So, what are the mechanisms for epigenetics to be accomplished? ازاي DNA بتاعنا او الجينيتكس بتاعتنا بتتاثر او الابيجينيتكس بتتحكم في الجينيتكس بتاعتنا؟ عندنا في main mechanisms DNA methylation, الهيستون موديفيكيشن والRNA interference، ودول بنسميهم الابيجينيتك كودز. اهم اثنين هما DNA methylation والهيستون modification. The DNA methylation of the cytosine in DNA occurs at the CPG dinucleotide. الحته اللي بيحصل فيها اسمها epigenetic mark. الفكره ان لما بيحصل DNA methylation it makes your DNA او nucleosomes more tight. بيبقى ماسك جدا، بيبقى صعب انه يبقى transcribed into messenger RNA and then into proteins. So it leads to silencing of the gene. This cartoon shows what I'm saying. When DNA is extensively methylated, it becomes an inactive gene and difficult for gene expression. What is important about this is that this change is reversible, and this makes it a therapeutic target. Another example of epigenetic codes is the histone modification. عندنا اللي شرحناه الهستون tail اللي بيبقى protruding from the nucleosomes. The site of many changes that can happen, or modifications that can happen, acetylation, deacetylation, methylation, demethylation, so on. The most important is acetylation and deacetylation of the histone tail. على العكس الميثايليشن اللي بيعمل تايب باكينج للدي ان اي وما يخليش الجينز اكسبريسد، الهيستون اسيتايليشن leads to ان هو بيعمل ريليسنج للجينز، ريليسنج للكروموسومز and so they can be more expressed, leading to expression of the gene. وهم عكس بعض. So this can explain why لما يكون عندي اثنين identical twins، واحد اتعرض لظروف مختلفة غيرت عملت ميثايليشن او اسيتايليشن، واحد ما تعرضش للظروف دي، why ده كان express a disease، والتاني اللي هو اخوه عنده نفس الجينز this disease was not expressed in him. اخر مثال مهم ثالث للابيجينيتك كودز هو المايكرو ار ان اي او السمول انترفيرنج ار ان اي. المايكرو ار ان ايز دول عباره عن 18 24 نيوكليوتايد سنجل ستراند. المهم فيه انه نون كودينج ار ان اي موليكيول، والمهم فيه ان هو repress the expression of the gene products at the transcriptional or post transcriptional. ده موجود في الانيمالز والبلانتس، بس عكس البلانتس هو موجود في الانيمالز مش محتاج، هو عباره عن سيجمنت من الار ان اي صغير، مش محتاج كومبلمنتري سيجمنت من الدي ان اي عشان يمسك فيه، وبالتالي ممكن سنجل ستراند اوف مثلا مايكرو ار ان اي يمسك لكذا نون كومبلمنتري ستراند من الدي ان اي ويعمل له سبريشن. So several microRNAs are circulating in the blood. زي ما انتم شايفين كده ادي microRNA it is a small segment of RNA يمسك للدي ان اي نون كومبلمنتري، ممكن واحده تمسك في اكثر من مكان تعمل سبريشن للجين برودكشن. ممكن to so many organs affection المايوكارديال والرينال والهيباتيك انجري. So to sum up احنا الفينوتايب بتاعنا بيتحكم في الانفايرمنت بالويست والتوكسينز والدراجز والسموكينج والبكتيريا والفيروس مع الجينوتايب together الاثنين مع بعض produce الابيجينوتايب اللي بيبقى عن طريق الميثايليشن او الاسيتايليشن ستون موديفيكيشن والمايكرو ار ان اي. حاجه مهمه جدا بتأفكت ده is our food what we take. الفود بتاعنا فريش، الجنك، الكان، الحاجات اللي فيها توكسينز دي كلها بتأفكت us affect the epigenetic codes عن طريق الميثايل جروبس او الاسيتايل جروبس that are produced in this food. طيب does it have an application in medicine and nephrology? Can epigenetics be manipulated or not? هو ده سؤال clinical مهم. It has applications in many fields. In hematology, in cancers, in endocrinology, in diabetes, and what's new is in nephrology. And there is flooding of data now about epigenetics being a new way to look at the kidney diseases, about being a helpful tool for better understanding in clinical nephrology. Let us start with acute kidney injury. Diagnosis of acute kidney injury اللي هو السؤال الشائع لينا دايما when to diagnose it as much as early as possible to save the kidneys. This could be a tool to help us before depending or relying على الكرياتينين واليوريا والحاجات الاوردينري to diagnose acute kidney injury. It is an essential tool for development of successful therapies. من الحاجات اللي استخدمت كتير وعليها ريسيرش كتير المايكرو ار ان ايز deregulation has been found to be associated with major kidney disease, glomerular, tubular, with the size of mesangial cells, podocyte dysfunction, proximal convoluted tubules, survival in AKI. And using urinary microRNA as specific markers for very early diagnosis, many researches were done على AKI, especially in ICU patients. Another example, renal calcium excretion and epigenetics in AKI. However, in ICU or non-ICU patient, this paper was a very interesting one. اتعملت على control ICU non-ICU patients that was found to be a very sensitive marker for diagnosis of AKI before the rise of the ordinary markers we use. So, therapeutic use of microRNA antisense may be beneficial if given early enough to ameliorate or even stop further injury in AKI, and this has been proved not only on mice but also the research is now going on women. مثال تاني الابلكيشن بتاعه في nephrology CKD and with the evidence of its involvement of progression of renal fibrogenesis in many aspects, especially كمان diabetic kidney disease اتعمل عليها researches كتير جدا. Several mechanisms and enzymes and epigenetic codes were studied. أهمها كانت histone modifications with the use of the pharmacological inhibitors of histone deacetylase inducing fibrosis, methylation, CpG methylation, silencing chronic progression of renal fibrosis, DNA hydroxy methylation, DNA methyltransferase, and this group of enzymes is the longest group of enzymes. Now they are being produced in the form of drugs and being tried in AKIs and many diseases. And these are examples of the drugs really now under trials dexamethasone, والazacitidine, والphenylbutyrate, والtriphostatin, in many drugs, sepsis-induced acute kidney injury, ischemia reperfusion injury, diabetic nephropathy, polycystic kidney, all these are under trials. In very near future, these would be drugs prescribed for treatment of such conditions. And in neurological malignancies even. The use command for dialysis patients has been related that these epigenetics codes incriminated in renal neoplasms associated with dialysis and with the hyper-responsiveness of some patients to erythropoietin expression are related to them. In transplantation patients also has application في early diagnosis of rejection, في long-term graft survival association, في infection with viruses and the early diagnosis of them, especially with CMV and BK viruses. So, can epigenetics be manipulated? In theory, up to now, yes, it can be manipulated with the drugs we have spoken about, with the inhibition of the mechanisms we have said, DNA methylation inhibitors, histone deacetylase inhibitors. But what is now الحاجة اللي منعينا أو الستوب لسه research إنها it lacks the target specificity. إنها ممكن to act over the whole body not in a specific site. But this was time هيبقى فيه more changes and more achievements. So my final take home message is epigenetics and epigenetic modifications will be the then part of modern biological science. We are still in infancy in understanding them, and further research needed to study the association between global DNA modification, gene level expression, and silencing in different branches of medicine, with the effect of various epigenetic interventions on disease outcomes should be studied. And I want to end with this nice cartoon describing how the disease progresses in our body, starting from the genetic and epigenetic association, be translated into biological knowledge and clinical implications. And then several steps now ممكن attack the disease at them. At the level of RNA, and these are transcriptomics, at the level of the protein produced by the RNA, proteomics, metabolites produced, metabolomics, biological functional studies. So now we have what is known as the genetic dissection of a disease. Genetic dissection. And the future will be for personalized therapy, epigenetic drugs, and self-prescribed targeted therapy for specific diseases. And finally, thank you very much. And I want to say, if you have, لو حد جالكم من ولادكم أو من أحفادنا أو أخواتنا, don't blame them for مش جايب نمرة كويسة في المدرسة. Don't blame them for themselves. Just blame your terrible genes. هي السبب راحت للولد وعملته كده. Thank you very much.
Thank you very much. One question? Just very quickly again for the young nephrologists. So if you have a disease, you have susceptibility genes predisposed to the disease, like polycystic, وبعد كده في modifier genes that determines the course of the disease. ودلوقتي زودنا عليهم manipulation and metabolites and metabolism of the gene through epigenetics. Thank you very much. Our next talk is management, software, API.